For lab this week, we're going to be doing osmosis, and we have two parts to our osmosis experiment. We have a plant and animal osmosis. For plant, we have an example of Elodea, which is our water plant, and for the animal, we have a chicken egg, and we look at osmosis in this two areas. What is going to be the order of activities during our lab? During our lab this week, the lab is going to be carried out in shifts. Your instructor will email you and let you know which shift is supposed to come at which time. During lab, we will have it start by making a wet mount of an Elodea leaf slide. So previously we had fixed slides. That's what we used for our cell structure lab. This time we're going to actually make this slide. So we're going to make an Elodea leaf slide. We'll get out our microscopes and observe osmosis in this plant slide. Then we're going to learn how to use a triple beam balance, because that is going to be our weighing mechanism for this lab, and your instructor will give you instructions for how the triple beam balance works. Then we're going to set up the egg osmosis experiment, and then your instructor will let you know which group you're in. Some groups will be using mystery solution A, and some other groups will be using mystery solution B, as we saw in the egg experiment videos. We start by defining what osmosis actually is. It's defined as the movement of water across the selectively permeable membrane. If then it is the water that is moving, in which direction does the water move? It's said that the water moves towards a region of higher solute concentration. Solute is the thing that you put in that dissolved inside something. So if you have sugar dissolving in water, then your sugar is your solute. So in which way does the water move? It moves towards the region of higher solute concentration, because the cells are always trying to achieve osmotic balance, and the water moves to help it do that. In the cell that we have over here, we see that in the middle there's a lot of solute, and on the outside of the cell, beyond the selectively permeable membrane, there is less solute concentration. So which direction do we expect the water to move? We expect it to move toward the region of higher solute concentration. So the water would move into the cell. Yay! We are correct. The water does move into the cell. Next, we would like to define terms related to tonicity, right? And when you talk about osmosis, you cannot use tonicity terms by themselves. You have to use tonicity terms comparing the cell with what is inside the cell with what is outside the cell, the concentration of two solutions. You can't compare the tonicity of one solution by itself. Therefore, a hypertonic solution is one that has more solute concentration compared to another solution. In our case, the inside of our cell, the cell itself. A hypotonic has a lower solute concentration. So you could say it's dilute, lower solute concentration compared to the other one. And isotonic is having the same solute concentration as another solution. So how does this behave in context of an animal cell and a plant cell when you have a hypotonic, a hypertonic, or an isotonic solution? In hypertonic solutions, these have less solute concentration compared to the inside of the cell. Then in which direction will the water move? It will move towards where you have higher solute concentration. So the water moves inside the cell. What happens to animal cells? When it gets too much water, then it explodes because it just has a cell membrane. What happens to the plant cells? When they get a lot of water, they become turgid, which means that they are the cell membrane and the cell wall are right against each other, and the cell is big, fat, and round. The isotonic solution, then the water moves in both directions and it's achieving a balance, whereas the hypertonic solution, so the solution on the outside has more concentration than the solution in the inside of the cell, then the water will try to move towards where there is more solute concentration, so it will move out of the cell. For animal cells, they will become small and shriveled, whereas for plant cells which have a cell wall, then you will see where the cell membrane will shrink, the cell wall stays in place, and it becomes plasmolized. And we see that in the videos that are provided following. Where we are preparing our Elodea wet mount, we'll get our young Elodea leaf we'll put it on a slide in a drop of water, we'll put a cover slip on it, and then we view, we draw what we see, and we answer the questions on the worksheet related to that. Then after we have made that fast drawing, we add 10% salt solution, which is very, very concentrated. It's high solute concentration compared to the inside of the cell. Then we view that, we draw and answer questions. So once we are done doing that, we want to put back our microscopes, clean the slides, and clean up our mess. So for the slide, the cover slip will go into the glass container, and then you will rinse your slide and put it at the edge of the sink, and your TA will take care of it after that. Then you'll clean up your table and make sure that you don't have any salt there and it's all clean, and then you will put away your microscope, and then we're ready to move on. Expect under the microscope to see something that looks like this, and this will be your chloroplasts, these green ones. If you're lucky, you will see them moving around in a cytoplasmic stream within the cytoplasm. So in the middle of the cell, within the cytoplasm, sometimes you have a cytoplasmic stream where they're just moving around and around. It's really cool to see. We know that the edge of the cells have a cell wall. Right against it is the cell membrane, but you're not able to clearly see the separation. So you know what's on the inside part is the cell membrane, and what's on the outside is the cell wall. So you can see the movement within a turgid LODR. So turgid, when it's turgid for the plant cell, it means that water moved into the cell, and you can see the cytoplasmic streaming, and we hope that you can see that in your videos as well. No, not your video, in your cell that in your slide that you're going to make. We can also see in plasmolysis, you can see that the solution outside is more concentrated, the water is moving out of the cell, and you can actually see how the cell membrane is going around the chloroplast and the cell wall is being left in place. And I'd like for you to draw that in your worksheet and then label where the cell wall is right over here and where the cell membrane is. The next exercise that we talked about was using the triple beam balance. And so this is the triple beam balance. This is where you place whatever you're interested in weighing. This is where your sliding weights, where your weights are, and this is where you see your balance. And we have three layers on the weight. The front one is in grams, the second one is in hundreds of grams, and then the last one is in tens of grams. And as we saw in the egg osmosis videos, then you can use this triple beam balance to weigh. So you will have your instructor will give you time, and you will go in groups to practice how to use the triple beam balance. Then I'd like for us to think about hypothesis related to osmosis within the egg. How do we expect the egg, or the weight of the egg, is going to change if we put it in a solution that is isotonic to the egg? We just saw water moving back and forth. So do we expect the weight to change? Do we expect the weight to stay the same? What about if we put it in a solution that is hypotonic to the egg, which means that that solution has less concentration, less solutes than the egg? In which direction is the water going to move, and how does that affect the weight of the solution? And finally, how would the weight of the egg change if you put it into a solution that is hypertonic to the egg, which means it's more concentrated, it has more solutes compared to the egg? In which direction is the water going to move, right? In relation to the egg itself, and what does that do to the weight of the egg? And so our mystery solutions, mystery solution A and mystery solution B, are related to these hypotheses and also what we start out with. So we want to be able to see osmosis in action, and the egg cell is big enough that we are able to do that. That we're going to work in teams of 2 or 3 depending on the lab. The initial setup will already be done by our teaching assistants. What they have done is that they have already put an egg inside a cup. They have covered that egg with vinegar for 24 hours, and it is covered in tin foil, and it is put on a tray that has your lab section. So if it is A1 or D1 or B1 or B2, the lab section has it's already there, and then they cleaned up their mess. So now it will be our turn when we come for lab today. This is what we need to do. So we need to go on to step 2. We're going to adapt an egg from those on our tray for our lab section. We'll label the plastic cup that has the egg. We'll put in our name, our name and our lab section, and we'll give ourselves a fun name. And then we'll take the egg, pour out the vinegar, and make our observations about what it looks like, and then rinse the egg under running water, weight on the triple beam balance as we saw in the video, and then we're going to return it to the cup and cover it with a mystery solution. The mystery solution could be A or B depending on what your instructor has assigned you to use. Next day, if possible 24 hours after you set up, depending on your lab time, you should come back, right? And the instructor will have shown you where you need to come back, and then you will do the next step. So you will get your egg that is either in mystery solution A or B. You will rinse it under running tap water. You will weigh it on the triple beam balance, record all of your weights, and then discard the egg in the provided container. After this, you need to wait. We're going to wait until all the lab sections have collected data, and then we will be provided with a data set for egg osmosis. What will we do with this data set? We're going to answer the questions and the worksheet that are related to this data set. We are also going to make an average, right? We're going to make an average for the weight of the egg after being in the vinegar, for the weight of the egg after being in mystery solution A, for the weight of the egg after being in mystery solution B. We will make identification of which of these solutions is hypertonic, hypotonic, or isotonic, and then you will submit your worksheet on Moodle. Because it has drawings. You will take pictures of them and put them on your document and upload all of them onto Moodle. Thank you so much for your attention, and I'm looking forward to seeing you in lab. All your instructors looking forward to seeing you in lab. Thanks.